Okay. So we're still thinking about the soil system, the idea of plants needing access to nitrogen, and that's in very large quantities compared to all the other minerals. Some plants have this amazing ability to form a relationship with bacteria in order to team up and get their nitrogen needs met quite nicely. So when we think about fixing nitrogen, what we're talking about is the idea of going from dinitrogen, N2, in the atmosphere. There's 80% of it that we're sucking in right now into our lungs. Bacteria have the ability to take that and convert it to ammonia. So prokaryotic only. All right, so we're talking about bacteria, not eukaryotic cells. So not, ay, um, not fungi. Okay, not fungi. You get it? Are you hearing me? Not fungi. If you discover a fungus that can do this, you'll probably get the Nobel Prize. All right. It's a big deal. So please understand, not fungi. I love mycorrhizal fungi and fungi in general, but sadly, no, they cannot do this. So far, it's only been bacteria that are known to do it. Although, they've been looking at ways to use synthetic biology and actually engineer this capability into other organisms. But so far, they're not there yet. They have this enzyme where, fungi and other eukaryotic organisms do not. Bacteria have this dinitrogenase enzyme. It's a very unusual enzyme, very powerful in its ability to take that triple bond between those two nitrogens and break that. That's a pretty huge undertaking. And they can do that. So that it's pretty amazing. So what we have going on here in terms of when we think about plants and bacteria teaming up to get this to happen, we refer to the bacteria that hang out with the plants as rhizobacteria. All right, so you see that a lot in the literature. Rhizo is something root-like or root. So what they're trying to say here is they're bacteria that hang out with the plant roots and are capable of fixing nitrogen. So here are some of the genera that you see often studied for this. Rhizobium, Bradyrhizobium, Azorhizobium. So that's why they call them rhizobia bacteria. And when you look at the root systems, there's generally some kind of nodules that you can see on the roots with the naked eye. They vary in their shape and size and color, but you can usually pick them out. The first time I saw that, this was way before I took a plant class, I thought the plant roots were diseased and I felt very bad for the plant. Little did I know it was just the opposite. They were really nice little sweet homes for the bacteria to hang out in and fix nitrogen for the plant. So we're looking at something like this for the nodules. They look like tumors. That's what I thought they were, some kind of tumor. I think you can kind of pick up the reddish, orangish hue on those nodules on the right there. And that's not color coded, that's for real. Some of them can look reddish like that, and I'll explain why later. And then some of them are bigger, some of them smaller. But if you were to slice that open and look at it under the microscope, you'd see thousands of bacteria in that tissue. So it's a little home that's created. It's all, it's plant tissue that morphs into that nodule, and then it houses the bacteria and lets them have a cozy little space in order to do their job of fixing nitrogen. So it's a good thing. If you dig up your plants, some of your plants, and you see that, that's a good thing. Okay, so these nodules are an important area. And like I said, the plant produces those nodules. They morph their growth so that they can create that little home. So let me point out, so these rhizobia bacteria are the group of bacteria that do this nitrogen fixation with plants. There are what we call free living nitrogen fixers that just hang out in the soil by themselves. They don't partner up with a plant, and they're also capable of fixing nitrogen. So it's not only that, so the bacteria don't necessarily have to be symbiotic with plants for them to do nitrogen fixation. So for example, Anabina is a cyanobacteria that can fix nitrogen. They're kind of special because they can fix nitrogen all by themselves. They can also hang out with certain aquatic plants and fix nitrogen. They can do photosynthesis. So it's kind of they're extra special in that regard. But I'm going to come back to this idea. They often look, so these are the vegetative cells, these, and then you see this weird looking cell that they've labeled a heterocyst. And that's the site where they're actually doing nitrogen fixation, not in the vegetative cells. So they create a special compartment, a special cell that then they devote to nitrogen fixation. And like I said, they can be free living. Sometimes they hang out with plants. Here's another image of that heterocyst, and I'll explain why they do that in a minute here. I'm sorry, my dog is really, really sick right now. She's kind of crashing. Okay. So I'm a little worried about her. I have an appointment right after this. Okay, so heterocyst right there is that site of nitrogen fixation. They don't look like much under the microscope. They tend to be rod-shaped bacteria. They sometimes have flagella, so they can move around, so that's good. But they don't look like anything special under the microscope, and yet they have this very special enzyme that's letting them fix dinitrogen. So here, this table is showing free living nitrogen fixation on the bottom. So there's a variety of different bacteria that can do this, different species. They're not just limited to one genus, for example, or one species. You can see there's a lot of options here being listed out. And then up top on this figure or table, you see that there are plenty of different genera. These are different genera that are listed out that hang out with different plants. And so you can see like Azolla is aquatic, it's a water fern. The Frankia species, they're really interesting because they can hang out with certain trees and fix nitrogen, which is kind of special. Elder trees, we have plenty of elder trees in this area. So if you ever see a elder tree, like if it gets windblown and you look at its roots, if its roots are uprooted, look for the nodules. And then legumes, there's plenty of leguminous plants out there that do this symbiotic relationship. Sugarcane, that's a huge agricultural crop, of course. So there's lots of different plant microbe interactions here for this, but we tend to focus on a lot of them are crop producing plants. So soybean, alfalfa, bean, clover, pea. That's where the lion's share of research has been going on. And if you do any kind of like Google search and Google Scholar or something, you'll see by now there's got to be thousands of papers published on the idea of this relationship. So they're very interested in this relationship, trying to understand it better. And then, like I said on Friday, the kind of the holy grail idea is, can we create crop plants that don't do this with a bacteria? Can we have them do it? And they are working on that and making progress, surprisingly good progress on that. So let's talk a little bit about this process. If you think about a plant seed, let's say it's a alfalfa plant, and it starts to germinate and grow, and it's in the soil. And then, of course, hopefully it's a nice healthy soil where there's lots of different bacteria present. You have to imagine a situation where, okay, that bacteria that can fix nitrogen for the alfalfa, they have to find each other. It's a very messy, complicated world in the soil. The roots and that bacteria somehow have to meet up with each other. And so there needs to be a way for them to communicate. And they do that through chemical communication. So this positive chemotaxis to the roots, that's a fancy way to say that the plant roots in particular are excreting out chemicals to attract these bacteria and tell them to come to me, come to me, come to my roots. And so the bacteria start to follow that concentration gradient of that chemical signal and hopefully will make it to the surface of the roots. So makes sense that they have to have find a way to find a way to have a way to find each other. There we go. Some of the signals that the roots release are homoserine amino acids, AA amino acids. So this is a special chemical that can get released. Pea plants are known to do that. There also are flavonoid type chemicals that get released by the roots, or isoflavonoids, which are actually pigments. They are forming different colors like on fruits and vegetables, some of the reddish, bluish, purple pigments that you see. That's due to flavonoids. But in this case, they're actually being used as a chemical signal to get the bacteria's attention and get them to come to the roots. So as that pea or alfalfa seed is germinating and growing, this is part of their life. They're secreting these chemicals, looking for their friend, their partner. Then what happens is hopefully the bacteria make it to the roots. And once they get there, they will start to secrete what we call nodulation factors. It's a chemical that will get sent to the root, which makes sense again that the bacteria tells the plant, hey, I'm here and I'm not an enemy. I'm your friend. Let's hook up, so to speak. So they have this idea of, you got to establish that this is a friendly interaction and that it's okay to take it a step further. So these nodulation factors will dock into a receptor on the root, and that's the signal to the plant, oh, the bacteria, they've made it here. Now we can really get going on a relationship. And one of the first things that they see happen is that as soon as the plant acknowledges, like, oh, okay, I know you're here, their root starts to change shape. Some of the finer root hairs where the bacteria are, they start to curl and they can sometimes branch. And they think that branching might be just to increase some surface area for the bacteria to attach to. But it's typically the root hair area, the very fine roots and root hairs that are doing this. And that seems to be as this relationship is progressing, that's like the next step after you see the bacteria there and have secreted their chemicals. So here is the idea of a root. A root tip in this case is probably a root hair that they've magnified. And you can see all the bacteria hanging out. And then the mock-up drawing up top, they're trying to show the bacteria in red there. Attaching to the surface of the root hair. You have to imagine those nodulation factors releasing, docking into the receptors of the root. The root goes, oh, you're there. Let's get going. And so I almost imagine like the root is curling almost like it's trying to hug and trap the bacteria to keep them there. And that's one of the hypotheses for that curling action, where it almost seems like it scoops them up. They think that that's on purpose so that they can keep the bacteria close, hugged close to them, and they won't get washed away or taken away somehow. And so seems to be the next stage after they've talked to each other, so to speak. So this is the nodulation factor. And I'm not gonna, this is just in case you were interested. And I just wanted to point out that nod factors, they have this part in green here, and then they bracketed it and put a small n there. What that means is that's a subunit that can be like multiple of those, and so there's slightly different nodulation factors. It just depends on what bacteria you're looking at. So it tends to be that way because when they do this research, they tend to see that it's a very specific pairing, like not any old nitrogen fixing bacteria can hang out with pea, for example, pea plants. It has to be a specific pairing. This bacteria hangs out with pea. This bacteria hangs out with alfalfa. And so it seems to be a pretty specific pairing. So that's why it makes sense that there's different chemicals. Each bacteria has slightly different chemicals so that they can tell them apart and make sure the right partner is going to get into the relationship here. Okay. So the host, the plant goes, oh, okay, I get this nodulation factor. It's docked into my receptor. Yep, I know you're there now. The way they do that, these receptors, they're called lectins. These are lectins. They're sugar binding proteins that are in the root tissue. And so those nodulation factors dock into these protein receptors, and they really, if it fits in and docks in successfully, then it's the right bacteria. It's the right nod factor. It's the right bacteria. If that nod factor doesn't quite make it into the receptor, it's a mispairing, then nothing progresses. The bacteria, I mean, the root pretty much doesn't hear the signal and doesn't hear that the bacteria is there necessarily. And so they won't, it has to be a specific pairing, otherwise they won't take it any further. So after, let's assume it's the correct pairing, the right species of bacteria to the right host plant. Then you get the bacteria, they'll actually physically attach to tends to be the root hair that they'll attach to. And then that curling action or the branching action by the root tends to happen right after that. So this is just a mock-up of those nod factors docking into the receptor. So this is the root. Okay, this is all root tissue here. All right. And so those chemicals are docking into the receptor, those lectin receptors. And the only thing I really want to get across on this, other than showing you the docking going on, that's how the root or the plant is going, oh, you're here and you're the right person or the right bacteria for the job. Then this arrow, arrow, arrow that you see coming into the cell, what that's trying to get across is that once you have that nod factor docked, it sends a signal into the cell. So that's how the plant knows that the bacteria are here and ready to go. All right, it's a signal cascade. The fancy thing is signal transduction. That's what they call it, signal transduction. You're just taking that signal and transducing it into the cell. It's a cascade effect. We'll talk about signal transduction a little bit later, but once that happens, it actually gets to the level of the plant DNA, and a lot of stuff happens where the plant goes, oh, all right, we're ready to form this relationship. I'm going to turn on all the genes I need to get this nodule to form and to start partnering up for what we need to do for the nitrogen fixation. So the next step would be, let's assume the rhizobia have made it to the surface of the root hair, and they're like, okay, we're here, let's progress. The next step is called the invasion. I wish they wouldn't have used such an aggressive term, because usually you think about a negative thing here when you hear the word invasion, but this is actually a good thing. This is the idea that the bacteria will enter into the root hair, but in a very controlled way by the plant. The plant is controlling this. When you look at it under the microscope, it looks like a fine thread of bacteria. And so they call it the infection thread. That's where it gets its name. Visually, I'll show you some pictures. So you have to imagine think about a long root hair, and the bacteria tend to be attached near the tip of the root hair. Now they need to get into the root hair, travel down the length of the root hair, and get into the root tissue proper. So, but they need to do that in a way that doesn't trigger the plant to defend itself. So it has to be not so aggressive. So what they do is they will gently break down the cell wall of the plant cells, the root hair. They'll gently break that down just enough for the bacteria to sneak in past the cell wall. Okay, so that's like stage one. They made it past the cell wall. Now they're bumping into the cell membrane. Would it be a good idea for those bacteria to breach the cell membrane and break it? What do you think? No. If they break that barrier, the plant goes, oh, you're a pathogen. Yeah, I'm going to try to kill you. So that's not what the bacteria want. So the plant and bacteria, so the bacteria bump up into the cell membrane, but they stop there. They make contact with it, and then they essentially push the cell membrane and invaginate it down the root hair. So it's like they're pushing the cell membrane ahead of them. And you see these bacteria end to end to end, like, ants on a picnic table, just one after the other, marching down the length of that root hair. And the whole time, the lead bacteria is just pushing that cell membrane and invaginating the cell membrane down the root hair. And so that's the infection thread idea. So the cell membrane invaginates down the root hair. The line of bacteria are right behind it, marching down that root hair. And it looks like a thread, so they call it an infection thread. And that's the invasion step, right? But it's a good invasion. It's a good thing. So that mock-up on top is trying to show this idea where you have a little bit of a breach there of the cell wall. And then they're trying to show, see how that's the cell membrane, all that black, where it's outlined in black there, and then the red inside. That's the cell membrane being pushed down the root hair, and you can see how the deep red bacteria here are just marching down one after the other, down the root hair. So for the bacteria, the goal is to get to the root tissue. The parenchyma cells of the root tissue, that's where they want to set up camp. And this is the part of the root that's starting to morph into a nodule. As this is all happening, you start to get that nodule forming. So these figures down low are excellent images of, they've used green fluorescent protein and other fluorescent proteins to label the bacteria, and these are root hairs. And you can see the bacteria just going right down the root hair, and then they're setting up camp down low. You can see they're in the root tissue, parenchyma cells, starting to set up homes there. And then this one is, you can see that black line in that root hair there. That's the bacteria marching down the root hair. Here's some other images that are quite nice. I think this is green fluorescent protein again with alfalfa. So again, they call it a controlled infection. It's being controlled by the plant, because the plant's like, yeah, okay, I'll let you come into my root hairs as long as you behave and don't disrupt my cell membrane. Sure, come on in. And then the idea here is to get them to the root tissue where the nodule is starting to form. Again, you can see the root hair is a little bit bent here, but you can see that one looks like it has a double thread. It's pretty fancy. So the goal for the bacteria is to get to the parenchyma cells of the root tissue. And so they call this next stage the release of the bacteria into the parenchyma area, the parenchyma tissue. So what happens here, again, it's very controlled. So they don't just break into the parenchyma cells and swim around in the cytoplasm. What happens is the membrane, that cell membrane of the root hair, starts to bud off, pinch off, and in each vesicle will be some bacteria. And then that ends up in the root cells. So they're surrounded by plant-derived membrane again. All right, so similar to what we talked about with the arbuscular mycorrhizal fungi, that they're always surrounded by plant-derived membrane. So we call that bags of vesicles, we call them vesicles, and that's, if you were to peer inside those vesicles, they have the bacteria. So they, newer terminology is they call them symbiosomes, these vesicles that contain these nitrogen-fixing bacteria. Symbiosomes to get at the idea that it's a symbiotic compartment, essentially. Some of the older literature will refer to it as peribacteroid membrane. So it just depends what year it was published. But both of those mean the same thing. They're talking about the vesicles. Then once they get into the parenchyma cells and you start watching what happens with the bacteria in the vesicles, all of a sudden, well, not, yeah, it's pretty dramatic. The bacteria start to get big. Physically, they just plump right up and get big. And so then they have to give that a special term. So they call it the bacteroids. So if someone goes, oh, look at those bacteroids, all they mean is the bacteria have reached the stage where they're really getting big. And so they've really enlarged their cells, and bacteria can do that. Bacteria can change their shape and size to a good extent. And this is really kind of they're maturing. They're ready to go now. Like, okay, now they've set up camp, they've enlarged, and now they can be this workhorse where they fix nitrogen for the plant. So then the last thing that happens is you need to get some really nice vascular connections. So the xylem and phloem has to hook up really nicely into those nodules, make sure that you get the bacteria well fed and well hydrated, but also it's a good conduit for the fixed nitrogen to get sent to the rest of the plant. So these vascular connections, it's a win-win. It will help the plant get better access to nitrogen. It's also feeding the bacteria that are in that tissue. And so the bacteria definitely get fed. The plant is willing to do that, so to speak. It'll give them a lot of carbohydrate. And then the bacteria are busy fixing the nitrogen. And so when you hear that word fix, that's in this case, it's that idea of taking the dinitrogen and they turn it into ammonia for the plant. So that in visuals is kind of what we're thinking about here, where they're trying to show how that membrane pinched off and you have the vesicles with bacteria inside. And then the lower image, they're trying to show that, this is all happening as the nodules are forming to house the bacteria. So if you slice this open, the image on the right, you can see there's a bunch of bacteria in that nodule. It's just chock full of bacteria that are going to start fixing nitrogen. Here's an electron microscope picture just showing how enlarged the bacteria can get. And so this is the symbiosome, and then you have that bacteroid inside. That's quite big for a bacteria. And so you have, again, that protection with the plant saying, yeah, you can come in, but you got to be in the membrane. And then you can see how big the bacteroids are, quite large. So, okay, you got your nodule, you got your bacteria inside, they're compartmentalized in these vesicles, you got your vascular connections all set up. Now they can start doing the reaction. It's expensive from an ATP perspective. For one dinitrogen to get fixed, they expend 16 ATP. So for the bacteria, this is not to be taken lightly. So I would like you to know the basics of the reaction. The idea that you're taking dinitrogen with 16 ATP gets you 2 ammonia. That's the main thing I want from that, but that's the full reaction pathway there. So when they look at this partnership and they go, how much is the plant sacrificing to get this to happen? What they do is they look at the amount of carbon required to give to the bacteria. So this is just to give you a little bit of an idea. They've done work with soybeans and they figure about 12 grams of carbon have to be given off to the bacteria in order for them to successfully fix 1 gram of dinitrogen. So this is just another way to say it's expensive. It's expensive for the plant to feed the bacteria, and it's expensive for the bacteria to actually run that reaction. And so it's not cheap. However, enough plants do this partnership that it seems to be worth it. The cost is worth it. The benefits outweigh the cost. So they're taking that dinitrogen gas and they're cleaving that triple bond. This is actually the nitrogenase or dinitrogenase enzyme. And you can see it's complicated. It's not a simple enzyme. It's multi-subunits. And ferredoxin, we're going to come back to ferredoxin. So I'm going to point that out now and we'll come back to it. Ferredoxin is very important in the light reactions of photosynthesis. So here it is showing up in this, helping this enzyme do its thing. So ferredoxin is kind of special. It plays multiple roles for the plant. Here, what this is trying to show is that ferredoxin, what the red means reduced. So that means it has its full complement of electrons. And what's happening here is it's donating electrons to the enzyme. And when you donate off electrons, then you become oxidized. So basically, this is showing that ferredoxin is giving an electron to this enzyme, and you have this electron transfer going on. And the end result through that process is to take the dinitrogen and convert it to ammonia. So this is not straightforward. It's this, you have to have all the subunits in play, everything functioning correctly. You have to have ferredoxin there ready to kick it off by giving off an electron, getting the short electron transfer, and then that provides the energy, the free energy for them to break the dinitrogen triple bond. You can see they're also pointing out the input of ATP to get this to happen. So they have to donate electrons plus push it with ATP in order to get this to happen. So it's significant. This image is trying to show this is a significant thing. There's something a little funky with this enzyme. Unfortunately, it's a complicated enzyme, but it's also, surprisingly, really sensitive to oxygen. If oxygen, if this gets bathed in oxygen, it quickly denatures and falls apart, which is not ideal. So this nitrogenase or dinitrogenase enzyme falls apart in the presence of oxygen, which means it's not going to function for the bacteria. And this partnership is like, oh, great, now we can't even do our nitrogen fixing. So that's not good, because most bacteria that fix nitrogen need oxygen. So that's a conundrum, right? I mean, it's like, okay, I need oxygen for my own ATP generating needs, yet my enzyme dies in the presence, or doesn't die, it denatures in the presence of oxygen. So this is not ideal. So they've come up with a solution to this. Through the process of evolution, there have been these heme proteins that have evolved that bind oxygen. So you have this going on in your body right now. You have hemoglobin that's binding oxygen and bringing it to your cells. They came up with this idea too, like, well, let's do a heme protein. We can bind oxygen to the heme protein. And then what they do is if it's bound up, then dinitrogenase enzyme isn't impacted by it. And so what they do is they have this heme protein and they'll release oxygen very in a very controlled way. So the bacteria still get oxygen to breathe, so to speak, but the enzyme isn't bathed in a bunch of oxygen, if that makes sense. So it's this idea that you can control the release of oxygen off this heme protein and make it a slow enough release that the bacteria are getting the oxygen they need to get their physiology going, but the enzyme isn't affected by it. They actually call this hemoglobin leg hemoglobin. The leg is referring to legume plants because it was discovered in legume plants. So they call it leg hemoglobin, and that's what makes those nodules look pink. Just like your blood is red because of hemoglobin, leg hemoglobin, those nodules will look pink often. It's pretty cool. And so this is the heme protein that's binding oxygen and then slowly releasing it off to the bacteria so that they can stay alive. And so there's different parts of this. It's a multi-subunit protein, and there's the so-called globin portion. And in the old days, well, they know that's produced by the plant. And then previous ideas, they knew bacteria have the genes for heme production, and so they thought the bacteria were making the heme portion of this leg hemoglobin. But newer studies are showing, no, the plant has taken that over and the plant does it all. And so you'll see conflicting evidence. If you go back in the literature, they say the bacteria make the heme portion, but now they highly suspect that it's really all due to the plant. Like the plant has taken that all over and they do it for the bacteria. But basically, once that all gets made, the globin parts, the heme parts all come together and it makes a fully functional leg hemoglobin. It binds that oxygen. and is slowly releasing it to the bacteria. The bacteria can take it up right away and use it. And then that keeps the levels around the dinitrogenase really low so that it can keep functioning and working. So it's a really effective mechanism to make sure that this enzyme doesn't denature and become useless. So this goes no matter what bacteria you're thinking of. If it's a free living bacteria, it still has to deal with this idea of its enzyme is sensitive to oxygen. And so with that heterocyst that I was showing you, that cyanobacteria that has that specialized compartmentalized heterocyst, what they do, if you were to crack open that heterocyst and look at it physiologically, there's virtually no oxygen in there. So they sequester everything in that heterocyst for nitrogen fixation. All the enzyme is there. Where's that image again? There it is. So in this heterocyst, no oxygen or tiny amounts, but the dinitrogenase is in there. Everything's necessary to fix nitrogen right there. No oxygen, and then the fixed nitrogen gets diffused out to the rest of the cells. So they structurally isolate where they do their nitrogen fixation. So it's not just the symbiotic relationship where they have to worry about too much oxygen. Any organism that has this enzyme, they have to deal with it. And I just put this in here to show you the similarity. So this is what we have. We have hemoglobin and myoglobin. And look at like hemoglobin. If you're taking BI218, the alpha helixes and beta sheets of protein configuration, you can see how similar they are. So it's pretty cool how you can see across different domains of life, they've come up with these solutions for binding oxygen. Okay, well, I guess we got to stop. We're a little over. So actually, I think the next topic is photosynthesis after this. We're going to get into that next. So please set up an appointment with me if you're still a little unsure on water potential before your lab report is due.